I want you to take a second and imagine the ultimate construction project.
Oh, absolutely.
Like not a skyscraper or some massive suspension bridge or something like that. We are talking about building a human body from a single microscopic cell.
Right, which is just wild to think about.
It really is. I mean, it is easily the most complex engineering feat in the universe. And it brings up this wild, almost, paradoxical question.
It isn't?
Well, how does a developing cell know whether to become a beating heart muscle or, say, a thinking brain neuron or just a piece of skin when every single one of those cells contains the exact same architectural blueprint?
Yeah, that is literally the fundamental mystery of human biology.
Yeah.
I mean, we all know that our DNA holds the instructions, right?
Exactly.
But simply having the blueprint doesn't actually build the building.
No, of course not. You need a crew.
Exactly. You have to know how to read it, which sections to unroll at which precise microsecond, and honestly, which sections to keep securely locked away so you don't build the wrong thing at the wrong time.
And that, right there, is the mission of our deep dive today. So we're looking at a really groundbreaking new paper published in the journal Nature, detailing something called the Human Development Multiomic Atlas, or HDMA.
This massive study.
It really is. Yeah. Today, we're going to meticulously decode the hidden grammar and the strict syntax of our DNA during fetal development. We're going to explore how certain proteins act as these, molecular foremen. We'll uncover the incredibly rigid physical rules of this genetic architecture, and then we'll see exactly how a microscopic typo made in your scaffolding before you were even born can cause a major disease decades later in your adult life.
Yeah, and the sheer scale of what these researchers have mapped, it's genuinely a paradigm shift. This isn't just some static list of human genes. It's a comprehensive, highly dynamic atlas of how the genome actually physically operates in real time to construct different organs.
Right. And why should you, our listener, care about this? Because this is the foundational blueprint of you. We are going to discover that the origins of the diseases you might face at age 50 or 60 were actually written into the microscopic spacing of your DNA grammar before you ever even took your first breath.
Which is a heavy thought, but so fascinating.
It is. So, okay, let's unpack this. If we're building a body, before we can even begin to understand the construction schedule, we really have to understand the materials on the site, right?
Yeah.
What are the basic mechanisms driving this fetal development?
Well, to understand that, we have to look at what's basically a biological trinity.
Okay, a trinity. Let's hear it.
Yeah, so it's transcription factors, chromatin accessibility, and gene expression. So first off, our DNA isn't just, floating around loosely in the nucleus of the cell.
Right, it's packed in there.
Exactly. It's tightly schooled around these structural proteins into a super dense complex called chromatin. And when it's tightly packed like that, it's completely inaccessible.
So blueprints are rolled up.
Rolled up, cemented shut, and the genes are totally turned off. So transcription factors are these specialized proteins that act like those master foremen you mentioned.
Gotcha.
They bind to specific DNA sequences, which are called cis-regulatory elements, to promote local chromatin accessibility. They literally pry the DNA open so the nearby genes can actually be expressed.
Okay, so sticking with our construction site analogy here, the genome isn't just a flat piece of paper on a table.
Not at all.
It's more like miles of blueprints tightly rolled up and locked inside these structural pillars. And the transcription factors are the specialized foremen walking the site, holding specific keys.
That's a great way to picture it.
So they bind to the locked pillars, those regulatory elements, and they unroll very specific sections of the blueprints so the cellular machinery can actually read the instructions and start building.
That is a highly accurate way to visualize it. Yeah. And what the researchers did with the HDMA was map out this entire construction site across human fetal development. They mapped out over 1 million of these unlocked blueprint sections.
Wait, over 1 million?
Yeah, over 1 million accessible cis-regulatory elements.
Okay, let's just pause on that number for a second. How is that even physically possible? If a fetus is incredibly small, and we're talking about unrolling microscopic blueprints inside individual cells, how do researchers actually see what the foremen are doing in there?
It's incredible technology. They used a technique called share-seq.
Share-seq. Okay, what is that?
Well, the highly technical term is a scalable, split-and-pool combinatorial barcoding platform.
Wow. Okay, yeah, let's translate that. What does it functionally do?
Basically, it allows scientists to capture both the physical chromatin accessibility, so what's open,
yeah,
and the resulting gene expression, what's being built from the exact same single cell simultaneously.
Wait, let me make sure I'm getting this right. They aren't looking at like one group of cells over here to see what blueprints are open and a totally different group of cells over there to see what materials are being produced.
Nope.
They are looking inside one single cell and seeing the open blueprint and the active construction happening at the exact same time.
Precisely. They essentially tag the cell's physical state and its active output with these matching molecular barcodes. And the sheer scale is what makes this a staggering achievement.
Right, because doing this for one cell sounds hard enough.
Exactly. I mean, a decade ago, profiling the internal state of a single cell was this massive, painstaking undertaking. But in this study, they profiled 817,740 individual fetal cells.
817,000, that is wild.
And these were taken across 12 different human organs, anywhere between 10 and 23 weeks post-conception. Through all this massive computational effort, they identified 203 distinct cell types operating on the site.
Okay, so a map of 1 million unlocked blueprints across over 800,000 individual cells. But here's where I get a little confused.
Sure.
If we have this massive construction site and it's unlocked differently in 203 different cell types, how do we know which specific foreman is responsible for which specific instruction? Like, if a regulatory element, the lock on the pillar, is opened, how do we link it to the actual gene it controls?
Right, because DNA is so long.
Exactly. A strand of DNA is incredibly long.
Yeah, and that is actually one of the hardest problems in genetics. Because a regulatory element is almost never right next to the gene it controls on the linear DNA sequence. It might be, tens of thousands of base pairs away.
Wow, okay.
So the researchers solved this using something called the activity by contact, or ADC, model.
Okay, what does that actually mean in physical space? Because if a foreman is standing on, say, page 10 of a stretched out blueprint, how on earth is he controlling the construction happening all the way over on page 5,000?
Because the DNA isn't stretched out linearly, the ABC model relies heavily on the three-dimensional folding of the genome.
Oh, right.
So imagine a massively long ribbon. If you lay it completely flat, point A and point B are miles apart. But if you crumple that ribbon up into a really tight ball, point A and point B might end up physically touching each other.
Okay, that makes perfect sense.
Yeah, so the ABC model predicts which enhancer regulates which gene by calculating two things. First, how accessible the regulatory element is, and second, its three-dimensional contact frequency with the gene's promoter.
So basically, they look at how active the foreman is and how often the section of the ribbon he's standing on physically loops around and bumps into the exact gene he's supposed to activate.
Yeah, exactly that.
That is wild. So the linear distance doesn't matter at all. It's the 3D physical proximity that drives the construction.
Yes, 3D space is everything here.
All right, I want to take a quick breather here to give you, the listener, a quick recap of what we've covered so far, just to lock it in.
Good idea.
First, DNA is tightly packed and locked away in vaults called chromatin, so it needs to be forcefully opened to be read. Second, transcription factors are the molecular foreman holding the keys to open these regulatory regions. And third, this amazing HDMA study mapped over 1 million of these active open regions across 12 fetal organs using that 3D looping method.
Perfect summary. But here's the thing. With one major catch, identifying the foreman and seeing the folded blueprints only sets the stage for the real breakthrough of this paper.
Oh, really? There's more?
Much more. Because it's not just about which transcription factors are present on the site.
Yeah.
It's about exactly how they are arranged on the DNA strand down to the absolute nanometer.
Right. So we are moving from just identifying the raw materials to understanding the actual architectural syntax, the actual grammar of our DNA.
Exactly. And to find this grammar, the researchers didn't just, eyeball the data. They couldn't.
Too much of it.
Way too much. So they trained deep learning AI models, specifically a tool called chrombpnet, to predict chromatin accessibility directly from the raw DNA sequence.
Okay.
And this algorithm was able to unravel a comprehensive lexicon of motifs. Yeah, these are highly specific letter sequences in the DNA that the foreman bind to. And what the AI found was that these transcription factors do not work alone. They cooperate to pry the DNA open, and they do so using two totally distinct types of architectural syntax, hard syntax and soft syntax.
Okay, let's visualize this because the physical mechanics here are just mind-blowing to me. When we talk about hard syntax, the study found a specific arrangement called a coordinator element.
Yes.
And it's like a highly choreographed molecular handshake, right? You have two different foramen, let's say a twisty one TCF4 complex and an ALX4 protein.
Right, those are specific factors.
And they have to bind to the DNA, but the absolute rule is they must sit exactly 5 base pairs apart, oriented head to tail. If they're 6 base pairs apart or if one is facing the wrong way, the handshake completely misses and the blueprint stays rolled up.
Yes, that incredibly strict spacing is the defining feature of hard syntax. It relies on direct, physical protein-to-protein interactions. The foramen actually have to reach out and interlock with each other to generate enough leverage to pry that dense chromate open.
Wow.
And the reason the spacing is so rigid actually comes down to the physical geometry of DNA itself.
How so?
Well, the study found that hard syntax motifs often require spacing of around 7 to 11 base pairs.
Yeah.
Now, why that specific number? Because 10.5 base pairs is exactly one full 360-degree helical turn of the DNA double helix.
Ah. So a spacing of roughly 11 base pairs means the 2 transcription factors end up sitting on the exact same face of the twisted ladder.
Exactly.
They are perfectly positioned to reach out and touch each other. If they're only 5 or 6 base pairs apart, one is sitting on the top of the ladder and the other is sitting on the bottom, completely offset.
You've got it perfectly. It's structural engineering on a billionth of a meter scale. But as I mentioned, hard syntax isn't the only way they operate. The AI also identified soft syntax.
Okay, so how does soft syntax differ? Does the exact spacing not matter as much?
Right, soft syntax is much more flexible. Instead of the foramen needing to physically interlock hands, it's more like a molecular game of tug-of-war against the nucleosomes.
And those are the heavy spools the DNA is wrapped around.
Exactly. So for soft syntax, the transcription factors don't need to physically touch each other at all. They just need to be in the general vicinity, anywhere from 20 to 150 base pairs apart.
Okay.
As long as they are both pulling on the same general region of the DNA ribbon, they cooperatively outcompete the heavy spool and keep the chromatin open.
So the architecture of our DNA relies on these incredibly rigid interlocking rules running right alongside these much more flexible collective effort guidelines.
Yes.
But wait, if hard syntax requires such precise spacing, I mean, down to the exact number of rungs on the DNA ladder, what happens if there's a microscopic typo? Like if one single rung is inserted or deleted during fetal development?
The entire localized system collapses.
Just from one typo?
Yes. If you change the spacing by even 1 base pair in a hard syntax arrangement, you throw the proteins entirely out of physical alignment. You completely abolish their cooperative binding. The chromatin slams shut, the gene turns off, and that specific cell lineage might fail to develop properly.
Which is terrifying, honestly, considering how many millions of times these cells divide.
It is a delicate balance.
Now, up until this point, we've talked exclusively about transcription factors that open the blueprints, accelerators to the construction project. But the deep learning algorithm found a massive surprise hidden in this fetal data.
Did?
Ubiquitous negative motifs.
Yes, this was huge. While the vast majority of the binding sequences discovered by the AI promote accessibility, meaning they unroll the blueprints, the model confidently identified a subset of 15 motifs that actively reduce accessibility.
They shut it down.
Right. They are repressor motifs. They're bound by transcription factor families like Zebi-snail, YY12, and NFY. And what was astonishing wasn't just that they existed, but that they were absolutely everywhere.
How everywhere are we talking?
Well, in some fetal organs, these negative motifs comprised over a third of all predictive motif instances.
Wait, hold on. Let me push back on that.
Sure.
Why would evolution waste energy building a biological system that actively blocks itself? You have this highly active construction site. You've gone through all the trouble of unrolling the blueprints and bringing in the molecular machinery, and then you permanently bolt a stop work sign right in the middle of it. That seems incredibly inefficient.
It seems inefficient until you think about the demands of a complex control system. These negative motifs are not just dumb roadblocks. They are universal brake pedals.
Brake pedals.
Or like a governor on a high-powered engine. In biological systems, having strong activators working simultaneously with weak repressors at the exact same regulatory site allows for exquisite fine-tuning.
So you don't want the construction crew building a wall at maximum speed forever, or they'll just build it right through the ceiling. You need a constant push and pull to maintain the perfect speed.
Precisely. The transcription factors are engaged in this dynamic equilibrium to maintain the absolute perfect level of gene expression. And the researchers actually proved this brake mechanism using human genetic data.
How do they prove it?
By looking at EQTLs, expression quantitative trait loci.
Okay, what exactly is an EQTL in this context? Let's just break that down for the listener really quick.
Sure. Essentially, these are natural genetic variants in the human population that we already know change how much of a specific gene is expressed.
Like natural typos we already track.
Exactly. And when the researchers cross-referenced these known variants with their brand new fetal motif map, the results were incredibly clear. If a genetic variant naturally disrupts a positive motif, gene expression drops.
Because you've broken an accelerator pedal.
Right. But if a genetic variant disrupts one of these negative motifs, like that ZBS nail motif we mentioned, gene expression actually spikes.
Because you've essentially cut the brake line and the gene expression just speeds out of control.
Exactly.
Okay, let me just lock this into place for everyone listening before we move on to the final and frankly most impactful part of this research. Just another quick 5-minute recap.
Go for it.
So we've established that the grammar of fetal development relies on precise molecular handshakes, which is hard syntax, and flexible tug-of-war pulling, which is soft syntax. And thanks to AI modeling, we now know that negative brake pedal motifs sit right inside active DNA regions to fine-tune exactly how fast our bodies are built.
Spot on.
Which transitions us perfectly into understanding how this fetal grammar actually impacts you and me right now, today.
Because we finally have the tools to look at what happens when this grammar is broken. So the researchers brought in GWAS data, genome-wide association studies.
And those are massive global databases.
Huge databases. They contain genetic variants that are known to cause complex diseases in adults. So they took these known disease-causing typos and mapped them directly onto their new fetal HDMA map.
And the overlaps they found are just mind-blowing. Let's walk through a specific case study from the paper, Coronary Artery Disease, or CAD.
Right.
Millions of people suffer from heart disease. Now, in the GWAS database, there is a specific genetic variant called RS127403774. It's literally just a tiny genetic typo. But the deep learning model revealed exactly what this typo physically does on the construction site.
And it's brutal.
It breaks a negative ZBS nail motif. It literally breaks the brake pedal.
Yeah.
And simultaneously, that exact same single-letter typo creates a new binding site for an activator protein called CEBP.
It is a devastating double hit to the system. You simultaneously lose the repressor, so no brakes, and gain an activator, so more gas.
Wow.
The gene expression in that area is going to absolutely skyrocket. And importantly, the researchers didn't just find this happening in, say, the adult heart muscle. They tracked this specific grammatical error to fetal muscle endothelial cells.
Endothelial cells, meaning the specific cells that line the blood vessels. Wait, let me process this. You're saying a single microscopic typo breaks the brake pedal. A&D builds a new gas pedal at the exact same time, and it happens in the blood vessels of a tiny fetus.
Yes, and what this suggests is that this tiny typo drastically alters the vascular function during fetal development. It literally lays down a structurally vulnerable foundation in the blood vessels before the baby is even born.
Oh, man.
And then decades later, under all the stress of adult life, that vulnerable foundation eventually collapses and leads to coronary artery disease.
That is incredible. The disease was locked in on day one. And it wasn't just heart disease they looked at, right? You had another fascinating example regarding asthma.
Yes. The researchers identified an asthma-associated genetic variant, but when they mapped it onto the fetal atlas, they found it altered a regulatory element, specifically in a fetal lung macrophage.
Which is a type of immune cell.
Right. Now, asthma is a disease that obviously plagues people in childhood or adulthood, but the vulnerable genetic region where this typo sits is only accessible and active during fetal development. Yet, in the adult lung, that exact section of the blueprint is permanently locked away in the chromatin vault.
This is genuinely mind-bending. Are we saying the die is completely cast before we take our first breath? Like, if this regulatory element, this lock on the pillar, is only open in a fetus, and then it shuts down completely after birth, how on earth is it causing an asthma attack when someone is 40 years old?
It comes down to cellular memory and lineage. Altering the chromatin state and the gene expression of a fetal cell permanently changes the entire developmental trajectory of that cell's lineage.
I see.
Those fetal lung macrophages eventually develop into the alveolar macrophages that populate your lungs for the rest of your entire life.
Okay, so the scaffolding might be removed in the adult, but the faulty wall was already built.
Exactly.
If those immune cells are constructed with a slightly faulty blueprint because, a foreman couldn't bind properly at week 15 of gestation, they will permanently overreact to allergens 40 years later. The blueprint may be closed in the adult, but the structural consequence of it misfiring in the fetus is completely irreversible.
The disease is literally a time capsule buried in the fetal epigenome. The grammar of our DNA is so deeply conserved and so rigidly enforced that a spacing error of a single nanometer during the first trimester echoes throughout an entire human lifespan.
Wow. So what does this all mean for us? We've journeyed from the tightly rolled blueprints of our DNA, mapping millions of active sites across fetal organs. We've visualized the rigid 5 base pair rules of hard syntax, where proteins must sit on the exact same face of the DNA ladder just to lock hands. We've uncovered the unexpected universal negative brake pedals hiding in plain sight to prevent biological overdrive. And finally, we've seen the haunting reality of how adult diseases, like heart disease and asthma, are actually planted as tiny typographical errors in our fetal architecture.
This paper really completely reframes how we view human development and disease. We are no longer just looking at a static list of biological materials. We have finally uncovered the syntactic rule book for how those materials dynamically assemble themselves into a human being.
And it leaves you, our listener, with a final, pretty provocative thought to mull over. This research shows us the precise hard syntax and the exact base pair typos that cause adult diseases during fetal development.
Yeah.
It makes you wonder, if we know the rules of this grammar so perfectly now, could the future of medicine be using something like CRISPR, not just to replace broken genes, but to subtly edit the physical spacing of these regulatory elements in utero? Could we essentially rewrite the punctuation of our DNA, fixing the spacing of a molecular handshake before a disease ever has the chance to manifest?
That is exactly the true frontier of this science, moving from simply reading the Multiomic Atlas to actively editing the architectural syntax itself.
I really want you to think about your own health timeline. Does knowing your body's foundation was built on such strict, tiny, ancient rules change how you view your own physical resilience? We started this deep dive by imagining the ultimate construction project, building a human body from a single cell. Now we know the success of that project entirely depends on the molecular foreman reading the grammar perfectly, right down to the very last letter.